I am now pleased to welcome Jack Goon.
Two forces are fighting on a battlefield. Soldiers from each side are intermingled and brawling, while a general looks on from a nearby hill, squinting to see who is fighting who. Cancer is a battlefield where T cells, the same white blood cells that help you fight off an infection like the flu, are fighting cancer cells. And researchers, we're like the general on the hill, trying to make sense of everything and design drugs to help T cells win. I study mouth cancer, and in recent years, we've had good success with drugs that block cancer cells' weapons, allowing T cells to clear tumors in around 20% of patients. But the other 80% don't see any benefit. Why is this the case? Well, cancer cells aren't alone. They recruit allies, so-called immunosuppressive cells, to fight by their side. We need to target these immunosuppressive cells, not just cancer cells. But what weapons are they using, and where are they on the battlefield? Researchers have been trying to visualize the cancer battlefield for decades. Traditionally, they do this by taking pictures of the tumor. These pictures show gene expression, where each gene gives us a little more information about the identity and weapons of the soldiers on the battlefield. So cells express thousands of genes to complete their complex mission, but these current technologies only image a handful of them. So we have an incomplete picture of each soldier. It's as if the general on the hill forgot his binoculars and can't tell who is fighting who, let alone which reinforcements to send to the T cells. So how can we do better? In my lab, we use a new technique called spatial transcriptomics to take a deep scan of the battlefield. The process starts with surgeons who remove a patient's tumor. That tumor is then treated with a chemical that freezes all cells in place. Then we cut a two-dimensional slice and take a picture. But it's more than just one picture. It's 5,000 pictures of the same tumor, each imaging a different gene. The resulting data is huge, and it's my task to conduct the data analysis. I start by identifying cells as T cells, cancer cells, or cancer allies. Then I create a map showing where each cell is and a report on the cell's activities and weapons, like you see on the right. So far, I've analyzed 3 patient samples, and I found that T cells are often surrounded by cancer allies. And these surrounded T cells are less active and more deprived of oxygen. So maybe cancer allies are choking T cells as a battle strategy. With more patient samples, we'll gather more data like this to inform cancer drug design. And eventually, we can map every single patient's tumor to provide them with the individualized intel and treatment that they need to be cured. Thank you.